Thank you very much. Those of you that were present at the Asia Congress of Autoimmunity in Singapore would have, whoops, I hit something, would have remembered perhaps a very animated question and answer session between myself and Yehuda there on the issue of vitamin D, and in particular whether you would want to supplement vitamin D in autoimmune conditions. And as I was putting together this presentation, I thought, well, let's take the words that we agree on, and then I'll focus on what we don't agree on. So these are from Yehuda's recent papers, and vitamin D affects the immune system at many levels by a number of mechanisms. Vitamin D has multiple immunosuppressant properties. On the whole, vitamin D confers an immunosuppressive effect. The possible involvement of vitamin D deficiency in the development of autoimmune diseases has recently gained interest. The impact of the vitamin D pathway on immune function, its influence on both autoimmune diseases and cancer, should be further elucidated. I agree with that 100%. There is just one word on that page which I believe is totally incorrect, and that is the word deficiency. And so this presentation, I'm going to explain how the vitamin D metabolism in man really works and why the concept of deficiency is incorrect. Well, the vitamin D is important because it's associated with a nuclear receptor called the VDR. The VDR is a nuclear receptor which is diagrammatically shown here as just one element inside a cell. You have a macrophage, here a nucleus, cytoplasm, a whole lot of things going on in the cytoplasm. If you want more details, you can look on Wikipedia or you can look in Nature Clinical Practice at this citation here. But the VDR is important both for adaptive immunity, the T and B lymphocyte function outside the macrophage, and it's particularly important for innate immunity. It transcribes cathelicidin, TLR2, and the defensins inside the macrophages, the main defences that our phagocytes have against pathogens getting inside the immune system. The important thing is that innate immune functions of the VDR are unique to Homo sapiens. In Homo sapiens, and only in Homo sapiens, there is one nuclear receptor, the VDR, which expresses genes for TLR2, as well as the cathelicidin and beta-defensin antimicrobial peptides. All of these are essential to the intracellular innate immune defences. Humans are not tall mice without tails. They have different numbers of TLR, toll-like receptors, for example. Humans have toll-like receptor 10, and a mouse has toll-like receptors 11, 12, and 13. In humans, TLR8 detects single-stranded RNA, while TLR8 in the mouse, we don't know what it does yet. The VDR of mice is not part of their innate immune system. You cannot study vitamin D receptor in mice, how it relates to the immune system, and expect it to translate to Homo sapiens. It doesn't work. Whereas in man, the VDR transcribes cathelicidin, which is the primary antimicrobial peptide, or the primary antimicrobial, which produces peptides that protect the phagocytes themselves, and TLR2, which is the primary toll-like receptor that detects pathogens inside the macrophages. There's no such homology in mice. In mice, there are other receptors. There are other genes that do that. Even the higher primates do not emulate the cathelicidin innate immunity of Homo sapiens. For more information, we have just written a chapter, Autoimmune Disease in the Human Metagenome, which is in the new textbook, Metagenomics in Human Biology. It's coming out next month. Now, here we have molecular dynamics of the VDR receptor of Homo sapiens, and there is a drug sitting in the binding pocket. This drug is Olmesartan. The vitamin D, activated vitamin D itself sits in about the same spot. The green molecule is Olmesartan. Now, if we look at the VDR of Rattus novedicus, The VDR looks basically the same, but there are a number of amino acids that are different. And in particular, the amino acids that bind to that tetrazole moiety in human beings, here on the left, do not exist in the rat. And you can see the tetrazole is just wandering around aimlessly instead of having the two hydrogen bonds that cause it to be an agonist in Homo sapiens. In the rat, this particular drug has no similar effect. Got to be very, very careful when using animal models and the VDR, because the VDR is a highly evolved mechanism. Well, the NIH Human Microbiome Project has been trying to identify just how many genes, and in particular, how many genes coming from a microbiota, bacterial and viral genes, exist in the human body. The estimate is about 25,000 human genes and about 1 million bacterial genes. Where do they exist? Well, some of them are in all the normal places. We saw them on the previous shot, the nasal, oral, skin, GI cavities, etc. The places we expect to find bacteria. But there are also bacteria inside the immune system, persistent infections. This is a transmission electron microscope photograph from 1989, Columbia University, done by Emil Orozco's group. And he has stained in, this is a juvenile rheumatoid arthritis lymphocyte, but it's also in the macrophages and monocytes as well as the lymphocytes. But there are regions of bacterial DNA which are stained with urinal acetate. There are these long, thin, extruded, I guess I could call them biofilms. They're only biofilms in that they're an extruded protein, bacterial protein, and then there is this big colony here living in the cytoplasm of this lymphocyte. This is the nucleus of the lymphocyte. The nucleus is also somewhat under stress, as you can see. When we look at them with light microscopy, then what we can see is this is a monocyte which has just started to explode from the pressure of the pathogens in its cytoplasm. You see its nucleus is still intact, throwing out these long, thin filaments which come from bacterial proteins. There's no homologue that produces that type of filament in man. If we look at even simple genomes, if we look at the microbiome of the mouth, the salivary microbiome, we're talking about hundreds, literally 500 plus species of bacteria have already been identified in the saliva of healthy individuals. This study was done by Max Planck. It was done a number of healthy individuals all over the world. There was more variation individual to individual than there was from country to country. And in particular, look at these. Up here, Yersinia, the plague, Porphyromonas, Neisseria, Streptococcus. The size of the text is approximately relative concentrations that were found. But a whole series of species that one would never expect to find in healthy saliva, but it's there. And you can only see it when you use molecular technologies. When you sequence the RNA that you find in the mouth and you match it up with the known genomes of known pathogenic species, or known bacterial species. They're not all pathogens. In a hip joint, this was a study done by Dempsey in 2007, and not surprisingly, a large number of the species were typical biofilm type species. Lysobacter is a gliding bacterium. Also Staphylococcus is another known biofilm formation. But look at this here, hydrothermal vent eubacterium, something which is not supposed to be in the human body and certainly is not going to be a contaminant in a clean surgical environment, was sequenced using DNA technology or RNA technology out of the removed hip joints during a hip revision arthroplasty. Many human metabolites are affected by the metagenome. And the sum total of all the interactions between each genome, each pathogenic genome, and the human genome gives rise to the totality of symptoms suffered during chronic disease. The genes from bug A plus the genes from bug B create the environment for disease X to manifest. And as another example, genes from bug C, D, E, F, and without bug G, because many of the species are pathogenic to each other, could very well lead to disease Y. So we are now beyond the postulates of Koch, where one bacterium or one species leads to one disease. Now that we understand at the level of individual proteins, individual enzymes, how the species are starting to interact, we can see that we have a really difficult problem to solve. The genomes accumulate gradually during life, incrementally shutting down the innate immune system. Genes from the accumulated metagenome determine the clinical disease symptomatology. And in many people, they don't end up with a diagnosable disease. They just end up with the aches and pains of the aging, maybe with a cardiovascular terminal event, or maybe something like dementia. So VDR. I said that VDR deficiency is a flawed concept. And the reason it's a flawed concept is because vitamin D is not a nutrient. A vitamin is defined as something that the body does not make. We've known for decades that the body makes its own vitamin D, and it doesn't need sunshine to do it either. It makes its own vitamin D within the cells that need vitamin D from a substrate 7-dehydrocholesterol via a number of other transcription factors. There's PXR, which is another nuclear receptor, and then there's P300ZPB, which is another transcription factor, creating enzymes that take the 7-dehydrocholesterol through a 2,5-hydroxyvitamin D stage, which is an intermediate that most of you would be measuring in your patients, to the activated form 1,2,5-dihydroxy, which then activates the VDR and allows the innate immunity to go forward. But there are feedback paths. Feedback paths from the activated VDR, feedback paths into the PXR as well. There are feedback paths, there are feed-forward paths. This is a tightly controlled steroid. It is not a first-order mass equation vitamin. It's not a first-order mass action vitamin. It's a controlled steroid. More information in my paper there, Vitamin D Discovery Outpaces FDA Decision-Making. The important thing to know is that downregulation is not deficiency. It's the body regulating the concentration of the metabolite. When you observe a lowered level of this metabolite in the body, it's because the body is trying to regulate it down to make the metabolism work better. I won't go into active and precursor D metabolites. My colleague, Dr. Blaney, has written on that, and I'm sure will go into that in the subsequent presentation. But there are some citations if you're interested. The important thing to note is that the vitamin D and the 2,5-hydroxyvitamin D, the precursors that you normally use clinically, actually inhibit transcription by the VDR. They actually are immunosuppressive. I agree 100% with Yehuda and his colleagues. Vitamin D, exogenous vitamin D, is immunosuppressive. And only the 1,2,5-dihydroxyvitamin D, or calcitriol, can actually activate the VDR, and it can only do it in a paracrine fashion within the cells. Well, look, if you were a pathogen and you wanted to persist in a patient, it would make sense that you would knock out the defences of the cells that you were living within. EBV is an obligate pathogen. We know it lives within the cytoplasms of the phagocytes. EBV knocks out VDR. By knocking out VDR, it allows itself, VDR is this one here, it knocks out VDR, and it allows itself to persist because it stops the body from producing its antimicrobials, its endogenous antimicrobials, and the TLR2, which is a key part of the defense recognition mechanism. EBV does that. And in particular, in these lymphoblastoid cell lines, the 18-month cell line and the 2-month cell lines, the VDR is downregulated progressively as time goes by. Another pathogen that knocks out VDR very effectively is Borrelia burgdorferi. That knocks down or downregulates VDR expression 50-fold with live spirochetes and about 8-fold with the lysed form of Borrelia. And of course the bacterium that Yehuda identified back in the 1970s as being at the heart of autoimmunity in some way, in his early papers, Mycobacterium tuberculosis. The way that this pathogen evades the immune system and can live within the phagocytes is it downregulates the expression of the VDR nuclear receptor, it downregulates the ability of the phagocytes to fight it. So, let's have a look at this paper that's just come out, last week actually, in Emerging Infectious Diseases, a CDC publication, and it showed vitamin D deficiency in tuberculosis. What it showed was that the mortality from tuberculosis increased, the mortality increased as the level of 2,5-D, the vitamin D precursor, in the bloodstream decreased. So if the body is not down-regulating the 2,5-D, then patients have a far better life expectancy, or far better, lower chance of morbidity from tuberculosis than if they have a low level of 2,5-D. Well, that's fair enough. But the thing is, if we talk about this as a deficiency, then the temptation is to say, well, let's intervene. Let's give the person some of this vitamin D to replace this deficiency. But as we agreed on that first slide with Yehuda, vitamin D, exogenous vitamin D, is immunosuppressive. We all agree with that, whether it's molecular biology or clinical medicine. So would we give exogenous vitamin D, which we know is immunosuppressive, to try and prevent TB from expressing itself, becoming a morbid condition? Well, I wouldn't. We have to remember that low serum 2,5-D is a result of the disease process and not the cause. The more serious the disease, the greater mortality, the greater the vitamin D dysfunction, and the lower the measured 2,5-D quartile. So then we come to autoimmunity. And we all agree that infection is somehow related to autoimmunity. We've had whole conferences here on autoimmunity in Budapest and subsequent to that. So if we wouldn't give it in tuberculosis, why are we considering that vitamin D supplementation might be useful in autoimmunity, where there's also likely an infective component? So what we did, once we understood exactly how the vitamin D metabolism was working, we chose immunostimulation over immunosuppression. We found a drug which happened to activate the vitamin D, and it's a perfectly safe ARB. When you change the dosing of it, it has a good action on the VDR, and it starts the innate immune system working again, to the extent that people that are normally taking large doses of antibiotics, the moment they take the VDR agonist, they become very sensitive to even small doses of antibiotics. It's immune stimulation rather than immunosuppression. And this is data that Dr. Blaney presented at Porto. No, actually it was Dr. Perez, Tom Perez, Captain Perez, presented at Porto, showing a whole range of autoimmune conditions and the positive responses that we have been achieving using immunostimulation rather than immunosuppression over the last 8 years. So the take-home points are that vitamins are substances which cannot be made by the body itself, but we know the body synthesizes vitamin D from 7-dehydrocholesterol, so therefore we shouldn't be talking about vitamin D as a vitamin. It's not. It's a gene transcriptional activator. It activates gene transcription. It's a paracrine steroid. Paracrine, because it's primarily limited within the cell in its activated state, it's a paracrine steroid. Ingested vitamin D suppresses the immune system, as do other steroids, and in the short term, patients feel better as a result of taking it. And that, I believe, is why Yehuda is suggesting that it should be a drug which is in the normal arsenal of a rheumatologist. I agree with that. It's a rheumatologist drug. It has to be determined by a physician whether it's the right thing to do. But in the long term, the ingestion of this steroid allows human microbiota to proliferate unhindered, ensuring the disease severity will progress year by year. That's all. Thank you very much.
Thank you, Dr. Marshall, for the very nice talk. Your presentation is now open for discussion and for comments.
Okay, thank you for my presentation. I'm Professor Marshall from Serbia, and actually 15 years ago we were maybe the first one who gave vitamin D analog in rheumatoid arthritis. It would have hesitated our work. So, but I'm still confused. After 10 to 15 years of a lot of work on vitamin D, still I believe, and even from your lecture, we do not make a difference between vitamin D and hormone D. When you say vitamin D, usually you think on 25-hydroxy, and that's one story. And this is a story that means nothing in the body. On the other hand, when you say D hormone, you think of 125, and that's the steroid hormone you are talking about. It's a totally different story.
That's correct.
Can we make such a conclusion you did just by saying vitamin D? You thought of 25-hydroxy.
I had this more complex slide which I skipped by. This has just been published last year in Annals of the New York Academy of Science. This shows, with a number of patients from the speaker who will follow me, Dr. Greg Blaney, the 125D that he measured, the activated hormone D, versus the inactive hormone D. We make a lot of distinction ourselves, but when I'm talking to an audience like this, I'm sorry I glossed over that because I was trying to put myself into your shoes rather than into the complex.
But maybe we shouldn't. I do as well when I do my talks, but we shouldn't, because that makes the people usual doctors do not make difference. And they try to improve some things by increasing 25-hydroxy dose you mentioned, and it's a mistake in many circumstances. Maybe sometimes hormone spores or analogs can be given to achieve something in tissues. We want to.
There is a difference between the active hormone and the analogs. Here I've got an example. This is the thyroid receptor, thyroid alpha receptor. And in its binding pocket, you can see T3, the 3 iodines of T3, and also 1,2,5-dihydroxyvitamin D, which has a very high affinity for thyroid. One of the reasons why is the vitamin D metabolism gets out of whack. Thyroiditis sets in, or certainly hypo or hyperthyroiditis. This is also from a paper that we published last year, my colleagues and I, Amy Crowell, and dysregulation of the VDR nuclear receptor may contribute to the higher prevalence of autoimmune diseases in women. So you may be very interested in that, because the reason we use an agonist is because 1,2,5-D is a very active hormone that does a lot of things in a lot of different body tissues, and it turns out that Olmesartan is far more focused in its action.